Welcome everyone to the February Jousse lecture. I'd like to once again thank Clash Orlando Lawyers again for sponsoring lunch for this month's lecture. Today we have the privilege of having Adam Velenosi present to us. He is currently a biobanker from Vancouver, Canada, who over a span of 7 years has been involved in biobanking. And currently, he is working at Praxis Spinal Cord Institute as the program manager of the International Spinal Cord Injury Biobank. So Adam discovered his passion for biobanking in 2014 when he was an intern at the BC Children's Hospital Biobank, where he was assisting in establishing the first pediatric institutional biobank in the nation. Adam has also held committee membership at the International Society of Biological and Environmental Repositories and is leading the network in global biobanking and biorepository community. So today, the title of his talk is International SCI Biobank, Catalyzing Scientific Discoveries in Spinal Cord Injury Research through the Distribution of Human Specimens. He has asked that if you have any questions, that we are going to save it for the Q&A period after his talk. But feel free to put it in the chat and I will get to it at the end of his talk. All right, so we now pass it on to Adam to give today's talk.
Perfect. Thank you so much, Brian. And thank you, Patrick. Thanks everybody for inviting me as well. I think I just need you to unlock screen share again for me, and then we will be off. Oh, it says somebody else is sharing screen.
Apologies, you should be able to screen share now.
No problem. Okay, can everybody see the full screen version of my presentation? There we go. Good. All right, so as you already know, my name is Adam Velenosi, and I'm the program manager of the International Spinal Cord Injury Biobank, or ISBE for short. And we're located here in Vancouver, Canada. The title of my presentation again is right there. So I'll start with a little bit of background in biobanking, and all of my slides are numbered. So if you do have any questions that you want to refer to a particular slide, please feel free to include the slide number in your comments in the chat as well, or note it down for the end Q&A session. So what is a biobank? A biobank is a facility for the collection, preservation, storage, and supply of biological samples and associated data, which follows standardized operating procedures and provides material for scientific and clinical use. In 2009, Time magazine named biobanks as one of 10 ideas changing the world right now. Why biobank? Well, with biobanks, there is a decreased mental and physical burden on patients, participants, and clinical staff. This is because only one consent form is needed to fuel multiple future studies. And furthermore, patients also only need to have samples collected once per given time point, since the biobank can divide individual samples into multiple small aliquots for long-term storage. A good biobank ensures high quality standardized specimens and data, which are processed by highly trained technicians, ultimately reducing specimen variability. There is also increased privacy, security, and trust with biobank. The requesting researchers never come into contact with identifiable clinical data, and all requests are evaluated by a neutral governing committee to ensure equity and transparency. I'll touch on that a bit more later as well. Biobanks catalyze and expedite research because they have an existing inventory of participants and specimens and data ready for researchers to request. And this is as opposed to a researcher recruiting directly to their own study, waiting for the participants, the samples, and the data to accrue before they can actually run any of their experiments. Requesting researchers save time and money on most of the operating costs since they don't need to set up their own recruitment and collections. Additional aliquots may be available and requested by the researchers years later, so long as the biobank's inventory is not consumed within that time. And lastly, most Canadian biobanks do not require co-authorship. Instead, a simple acknowledgement is all that we may ask for. So this next graphic was created by Amanda Rush under the supervision of a friend of mine who operates a biobank in Sydney, Australia. It depicts the foundational pillars of a biobank. So these include business strategy, governance, facility design, ethics, consent, quality management, data management, and sample processing. And what I really like about this image is that Amanda captures the concept that a biobank cannot have integrity, quality, and sustainability without a strong foundation in every single one of these pillars. And it is this complexity that's one of the things that really attracted me to the field of biobanking in the first place. As somebody who has had hands-on experience in sample processing, patient recruitment, data mining, database development, study setup and coordination, biobanks require diligence and attention to detail in order to be successful. There's always something that can be improved as research and technology advances. And so there's continuously at least one process that can be amended or performed better when you are a biobanker. And now on to ISBE more specifically. So ISBE is, as far as we are aware, the first polyuser SCI biobank. This means that all materials and their associated information are made available by request to researchers globally, provided that the proposed research has undergone ethical review and that it is aligned with ISBE's mission statement. A polyuser biobank is different from a monouser or an oligo-user biobank, which are intended for use by a single researcher or a single institution. ISBE's mission is to advance the understanding of the biology of spinal cord injury through the study of human biospecimens. And so a paper published by Dr. Francesco Marincola in the Journal of Internal Medicine stresses the importance of this mission, where he states, translational research is caught in a feedback cycle whereby complex multifactorial disease is confronted without sufficient understanding of human pathophysiology. And so this is somewhat true in spinal cord injury, particularly, as you can imagine, most research is, or a majority of research is conducted in animal models. And so without us having the ability to confirm and validate some of the discoveries and findings in human specimens, we're sort of stuck in those animal models. ISBE's vision is to serve as a resource to the global SCI research community. And ISBE's objective is to support research into the biological responses of human spinal cord injury. Here's a diagram depicting ISBE as a custodian or a gatekeeper between participants and researchers, ultimately leading to new discoveries in spinal cord injury research. Now, as I briefly mentioned earlier, a good biobank has a well-defined governance structure. ISBE's governance structure includes two main committees seen here in blue, and these committees ensure that we operate equitably and transparently. So the first committee is our Executive Advisory Committee, which is responsible for the strategic direction and supports the overall function of ISBE. I'm sure many of you today will recognize some of the faces of our EAC members here. And the second is our Operational and Scientific Access Committee, which is responsible for the management and oversight of our biospecimen requests and sample distribution. So all ISKIP committee members participate for a 3-year term with the possibility for extension, which is actually approaching later this year. We would love to diversify our committees further. So if you're a scientist and you're interested in joining our OSAC to review all incoming ISKIP requests, or you're interested in helping us strategize the Biobank's future direction as part of our executive advisory committee, please send me an email after this and we can continue the conversation from there. The CEDARNet Biobank Certification Program. So on October 2nd of 2020, ISKIP became a CEDARNet certified biobank. This is an internationally recognized biobank certification program, which enhances a biobank's reputation through the implementation of biobanking best practices. And again, this is what builds the confidence in biobanks. So the creation of 40 unique SOPs and policies were involved in this certification process, and we're constantly amending these as our processes evolve day to day. On to inventory. So ISKIP's biospecimen sources include the transfer of remnant materials and data from 4 of Dr. Kwan's legacy clinical trials with participants' informed consent. Now, at the time that these were transferred to the biobank, it was referred to as the SCI Biobank, which was designed as an oligo-user biobank intended for sharing specimens within UBC I-Cord only. Fast forward to 2019, and the International SCI Biobank was established, which functions as the poly-user biobank that we have today. This is also the protocol for which we are able to collect postmortem spinal cords, but I will explain a bit more about that later. In the left corner here, we also have our SCI imaging and histology study, which I will also provide some more details for later. Finally, we currently receive remnant biospecimens from Dr. Kwan's CASPER trial, as well as the RESET AXR 204 trial, which at this time is our only source of specimens from individuals living with a chronic spinal cord injury. We would like to encourage more projects to contribute remnant material and data to ISKIP's growing inventory, and I am available to assist with building the biobank's consent language into your informed consent forms, ethics applications, so please contact me if you're interested, as this is something that works best if we are involved before a study is fully approved, rather than having to reconsent a bunch of patients or change your consent forms in the middle of a study. Once again, these are just the study sources of our fluid biospecimens. You can find more information about them on our website, including the links to their clinicaltrial.gov pages by clicking the description on our website. Now, as I alluded to earlier, we do procure and store formalin-fixed paraffin-embedded spinal cords as well. Currently, this enrollment and collection is exclusive to individuals who decease following an acute spinal cord injury at Vancouver General Hospital. However, we are now working on expanding this enrollment and collection to individuals who decease with a chronic SCI in BC. Beyond this, we also hope to eventually expand enrollment and procurement to other SCI care centers around the globe. So this diagram illustrates the workflow of ISKIP spinal cord processing. We begin with the procurement of a full or partial length spinal cord that you can see here. And then we identify the segment which contains the individual's unique epicenter of injury using their premortem clinical MRI. So that can be seen here depicted in the red E as the epicenter. Depending on the length of the spinal cord, we cut additional segments rostrally and caudally until the entire cord has been segmented. And each segment is then transferred to the UBC MRI research facility, where they undergo advanced 7 Tesla MRI, and before being partially encased in a 3D printed mold and divided further into small blocks for paraffin embedding, which you see here in the middle. Now, the resulting blocks then become the substrate for our future ISKIP tissue requests. And so we only ever release sections or scrolls, not the actual blocks themselves. These images depict our spinal cord processing in a bit more detail. Here are the molds or a photo of one of our segments that is inside of its 3D printed mold. And you can see the slots here. So these slots assist in the cutting process. So when we cut these, we are trying to line them up as best as we can with that 7 Tesla MRI. And so that keeps us on track for future histological studies that might need to compare to MRI. Select blocks are then chosen and stained with ISKIP's core stains. And the stains are also reviewed and rated by our neuropathologist here at UBC ICORD. And that helps us essentially determine the damage of each block so that we can better select which blocks should be sectioned for different research projects. The stain slides are then scanned at 40 times magnification, and all digital images produced by these scans are also available for researchers to request. If possible, we encourage researchers to upload their own digital images of stained ISKIP sections after they have published their work. So this is not only to contribute to our growing digital inventory, but it also reduces the need for repeat staining across the same blocks in future requests, especially given how precious these samples are. Here's a snapshot of our current spinal cord inventory. We have 12 cords, and the green cells represent a segment which is currently in its block form and ready for biobank requests. The red cells, however, represent segments which are still currently in formalin. And so we prioritize the epicenter, the rostral 1, and the caudal 1 segments first. Those are the ones adjacent to the epicenter of injury. And the reason for this is that most of our research requests have highest demand for those particular blocks and segments, which is no surprise. We then also prioritize the caudal-most segments here at the bottom. And the reason for that is that many researchers will also use some sections from those distant segments for internal controls for some of their epicenter staining. We continue to work towards getting these remaining red segments out of formalin and into paraffin throughout the remainder of 2022. Now, I want to quickly provide a bit more detail about the SCI imaging and histology study that I mentioned earlier. So our UBC ICORD co-investigators, seen here at the bottom, utilize our spinal cord segments for MRI and histological staining. After the scans are completed, all of the segments and slides are actually returned back to ISKIP's inventory with an enriched data set. And then the team here strictly analyzes the resulting digital images. So in 2021, I amended ISKIP's ethics and protocol to include the storage and release of digital materials. This includes those high magnification scans of the stained tissue sections, which you saw earlier, as well as those 7 Tesla ex vivo MRIs that are obtained from each particular spinal cord segment. And so the hope is that this digital inventory expands our user base, opening us up to digitally focused research requests, as this is an area of research that I foresee growing dramatically in the years to come, especially given all of the advancements in artificial intelligence and machine learning based research. Here are some examples of the detailed images that we can obtain using 7 Tesla MRI. And for those of you who have seen a standard clinical MRI for comparison, those are typically about 1.5 Tesla, or in some rare cases, 3 Tesla, whereas these are 7 Tesla. So you can really see the difference in resolution, especially when you're looking at these cross sections, and you can differentiate between that white and gray matter in the cord here. So unfortunately, we do not have approval to store and release a participant's clinical MRI for biobank requests. However, I am excited to share that Praxis is currently in the early stages of establishing a clinical imaging repository for use in future research. So once this is established, we aim to eventually link our biobank spinal cords and the 7 Tesla MRI to those images in the repository, which we hope will open even more possibilities in spinal cord injury research. And here is an example of a 40 times magnification Luxol Fast blue stained spinal cord section. Iskip is currently storing this as part of our digital inventory. And usually these are in much higher resolution, but I couldn't possibly include a 1 GB file in a PowerPoint presentation. But you can imagine that these are essentially what we give for what we would give for our digital materials. To summarize our inventory, Iskip has enrolled 242 participants. We have collected 12 spinal cords. We currently have 888 blocks available for sectioning. We have over 17,000 serum aliquots, over 4,000 plasma aliquots, over 18,000 CSF aliquots, and over 600 PAX gene blood RNA tubes for downstream RNA extraction. So we haven't actually done too much with those PAX gene blood tubes yet, but given future biomarker studies, we are looking to eventually start extracting the RNA for those, at which point we can distribute RNA. We also have hundreds of those pre-stained digital tissue sections that I just showed you. And again, we encourage researchers to send us their digital images after they have published their results. Now, moving on to data. As I mentioned, data is one of those foundational pillars in biobanking. And so we are currently in the process of migrating from a Canadian-based database called ATOM to a more sustainable database called Open Specimen. So we selected Open Specimen because it integrates with platforms such as REDCap and many popular specimen labeling software. It can perform biospecimen and data queries on demand or automatically at predefined schedules. And then these can be shared directly with other users so that they don't need to repeat the queries themselves, which is pretty useful. And it also allows for admins to create controlled access user accounts. So in other words, if we want to provide limited database access to one of our international enrollment sites, we can set that user's account to only be able to view, edit, and add data for their specific site while remaining blind to all of the other data that is collected and stored in Open Specimen. And this is really helpful when you have enrollment sites scattered around the globe. So here are some screenshots of the minimal data set that we collect in each system, ATOM and now Open Specimen. And in both of these databases, we strictly collect a minimal clinical data set for querying purposes. But when it comes time to browse our inventory when it comes time to browse for more detailed information, we can then go to find additional data so long as we have access to the full clinical data set that was collected with the original study. But essentially, this minimal clinical data set we determined was all that was really needed based on research requests, just so that we could browse our inventory and be able to select biospecimens for particular researchers. We have also designed Open Specimen to have predefined visits and collection templates, and these are unique for each of the clinical studies that is affiliated with Iskip. Open Specimen also has the ability to automatically generate all of our labels based on the token that we specify. And this greatly reduces the chances of human error, especially when compared to manual data entry or, even with barcode scanners, those can get quite finicky sometimes. And so this not only saves a huge amount of time, but also maintains that integrity of the data and the accuracy of the data, which is so important in biobanking. And here's an example of the fully customizable storage containers that we use. So we have a freezer divided into shelves, then racks, then trays, and then boxes. And here's a box map that we use to quickly view our inventory locations without actually needing to open up a fridge and go in there or compare to, Excel spreadsheets, let's say. So we aim to fully adopt Open Specimen and complete all of our data migration by the end of the next fiscal year. Now moving on to our requests. The process of requesting ISKIP materials has also been standardized, and ISKIP is open to receiving requests from both academic and commercially affiliated researchers. So both processes begin with an initial consultation with Dr. Kwan and myself, typically initiated by email or now through our new website. And this allows us to determine the feasibility of using ISKIP specimens in your project before you actually submit a formal request. If necessary, you may need to amend your study or your ethics application to ensure that you are approved for research using human specimens. But once all of that is complete, we then have you submit a standard biospecimen request form and return it to us, at which point we will forward that to the Operational and Scientific Access Committee for review. Now, if approved, a material transfer agreement is reviewed and executed between institutions or parties. After that point, a test shipment of up to 10 specimens is then couriered to your lab for piloting your experimental procedures. And if successful, subsequent shipments may then be released. And the reason for this stepped approach is that it allows us to conserve our precious inventory just in case your experiments using human specimens are not successful. Because you have to remember, a lot of our requests are coming from academic animal model labs. And so they may not have the expertise, or maybe what they have discovered in animals is not at all relevant to human specimens. So while we have not yet released any materials to industry commercial users, we are open to it. However, a unique MTA must be developed on a case-by-case basis, and we would also expect higher fees to be charged for these particular samples. When receiving requests from an academic researcher, however, we strictly charge on a cost recovery basis, and sustainability is a whole nother conversation, but the cost recovery structure of a biobank only makes back about 20%, give or take, of the operating costs of a biobank. Now, to date, ISKIP has received a total of 20 requests and inquiries, 4 of which of these came from within our own institution at UBC. The rest of the requests have come from the United States, Canada, England, Germany, Sweden, Belgium, Portugal, and Australia may be a bit premature to include here, given that we just recently initiated the conversation, but I was excited to put some more color on this map. So there it is down here. In terms of outputs, ISKIP was recently acknowledged in 2 high impact factor journals. The first was in Nature Communications from Dr. Christian Goritz's lab at the Karolinska Institute in Sweden. And the second was in Brain Communications from Dr. Ravi Iyengar's lab at the Icahn School of Medicine at Mount Sinai in New York. As I mentioned near the beginning of this presentation, there is no requirement for ISKIP co-authorship in any of these publications. But we do have some standardized language that we ask you to include in any and all presentations and publications. And this is specified in our material transfer agreement. And The major reason for this also is that it really helps us track the publications more easily, because not everybody can remember to email or notify us when they do publish. If you're interested, the links to both of these papers can be found in the news and events section on the homepage of our website. Now, I will wrap things up with future directions here in just a couple more slides, and then we can open it up to questions. So what's next? I've organized our future directions by the categories of foundational pillars of biobanks that I introduced at the beginning of this presentation. So business strategy, to gain support from more institutions globally, improving ISKIP's long-term sustainability. Governance, to invite new members to join the Executive Advisory Committee and the Operational and Scientific Access Committee. Facility design and quality management, to install new dynamic environmental monitors in ISKIP's storage rooms and freezers, further protecting our specimens and ensuring that their quality is always maintained. Ethics and consent, to set up recruitment and collection workflows for individuals with chronic spinal cord injury. And we actually just had made some really good progress with this through some discussions that were held last month. Data management, to complete the database migration from ATIM to Open Specimen, which you saw earlier, by the end of the next fiscal year. And finally, sample processing. So to expand spinal cord procurement to other SCI centers globally. Now, while we understand that the processing is quite complex, we envision that ultimately the specimens would have to be shipped back to us here in Vancouver. However, our hope is that we can really get other SCI care centers to enroll participants for the spinal cord donation, and then have a trained morgue diener and collection specialists to be able to retrieve that precious cord without actually damaging it. All right, so I'd like to thank all of our supporters and affiliates that are seen here. This program would not be possible without them. And I'd also like to thank all of you for attending this virtual presentation. I hope that I've succeeded in teaching you all something about ISKIP and about the importance of biobanking. For more information, I encourage you all to visit our website at www.sci-biobank.org. We have a fairly comprehensive FAQ page on there as well, in case you don't get around to asking any questions that you might want to know the answers to today. Or you can always contact me at my email here. And at this time, I think I'd like to open it up for questions. And maybe I'll just leave this slide over here while we ask some questions.
Hey, thank you very much, Adam. That was very fascinating and a great overview of the biobank and how it works. So I'd like to open it up now for any questions that you may have. Feel free to put up your hand or to write your question in the chat or unmute yourself with a question. So while you're thinking over your question that you may ask, I do have a question, and it's always on my mind in terms of sustainability. And you did mention it very briefly about your business strategy, and that being that to gain more support with different institutions. And I want to see if, or I want to ask whether you could sort of expand on that, how that would help in terms of sustaining the biobank.
Yeah, for sure. So yeah, and in the past, I've also given a presentation on sustainability quite a few years ago. So hopefully I can do this some justice here. So as I mentioned, sustainability of biobanks are quite a difficult thing. Even with a cost recovery model, the challenge is that you're always collecting samples in in a quantity with a large overhead cost. And at the point of actually releasing those specimens, you're releasing a minuscule fraction of the overall cost that it took to generate and store that specimen. And so, like I said, cost recovery makes back about 20% is even a conservative amount. I mean, most biobanks are lucky if they can do 15%. There's a couple that I've seen do 25%. But it sort of stresses the point that you need other sources of funding. And so if you're unable to get core funding through an institution, like thankfully, so far we've been able to set up and operate through donations made by Rick Hansen Foundation and Practice Spinal Cord Institute. But on top of that, biobanks also have an opportunity for outsourcing other services. So this is not something that we really do at UBC ICORD, but for example, if a biobank has specialized equipment like a blood fractionating robot or like a slide scanner or a DNA extraction robot, you can sort of charge other labs on a unit by unit basis to try to recover some of those costs. But at the end of the day, the largest expense is really your staff. And so we've been really lucky that Dr. Brian Kwan has such an ambitious mind and is able to apply for all of these grants and be able to successfully obtain grants where we can, given that they overlap with the biobank. So for example, like I mentioned, that Casper study, we're able to sort of split salaries across different projects and try to subsidize the operating costs of a biobank as much as possible. But for our particular biobank, which is very unique, obviously, those spinal cord collections. So, I mean, without even factoring in any technical time and without factoring in technical time, it's pushing, $6,700 to $7,000 a cord. And so it is quite expensive. Thankfully, that is now being covered under a grant, which was obtained by the co-investigators that I showed you with the SCI imaging and histology study. So we really are trying, or we're doing okay right now for the fact that we have we have grants coming in from multiple sources that support the biobank. But the reality of the matter is that a biobank essentially needs some kind of core funding from institutes or from federal and provincial government in order to really remain sustainable long term.
Thank you very much. Yeah, funding is on the top of mind of many researchers and organizations such as yours. Are there any other questions that people may have? Again, feel free to put it in the chat or to raise your hand. While we wait for that, I have many more questions. I guess sticking on the topic of sustainability as well. I know it says international in the title, but it's most of the work seems to come through BC, and a lot of the people in your committee are North American in terms of where they originate. But then this may be a silly question, but is there an opportunity to, I guess, expand the organization so that and I think you alluded to that slightly in terms of where to keep samples and whatnot, but to expand it internationally and maybe spread out, or at least have mechanisms to enhance sustainability in that way?
Yeah, so this is something actually that was proposed in the early draft of the business plan. And so there's a couple of benefits to that as well. So if we were to create, maybe a secondary site or a satellite site, it would definitely split the cost for facility management and technical time and things like that. Furthermore, it also kind of protects our samples in case there's, perhaps a natural disaster and the samples get destroyed. That's another issue that you would potentially face. And so we never really had anybody jump or volunteer in that opportunity just yet. I mean, not to say that it might not happen. I think that it is a lot more feasible as well now that we are migrating to open specimens. So as I mentioned to you, we can kind of divide access on a user and site basis. And so one of the biggest concerns that we had about potentially dividing the bank into two different sites, like maybe having a European center, the challenge was that, well, how are we going to be able to actively see the live updates for all of the samples, and to be able to, given that Brian is using his using open specimen also for Casper and other things, how can we restrict that? And so it wasn't really possible with Iskip or with ATIM, but now that Iskip is moving to open specimen, part of the reason that I decided to make the move to open specimen was having that in the back of my mind as well. So I think it's a possibility. I'm open to it, and I think even in terms, like I said, of protecting ourselves from natural disaster, for example, it would be a really good idea to potentially divide those samples into two different biobanks under the same umbrella name.
Yeah, no, that's a good point. I didn't even think about physical space and how there's some dangers with one central location. Yeah, that's a good point.
That's one of our SOPs, actually, which is like disaster.
Yeah, oh, I can imagine. So we have a question from Caitlin in the chat, and I'll read out the question, but Caitlin, you may want to unmute yourself to maybe elaborate on your question a little bit more. But the question here is, do you supply samples for SCI-related conditions? So that's the question. I don't know if you want to elaborate on that more, Caitlin.
So sure. Yeah, so we're doing an experiment involving, or a study looking at degenerative neuromuscular conditions. So I was just wondering, like, we're looking at wanting to get healthy CSF aliquots, but obviously that's very difficult to come by. So I was wondering if we could get CSF samples from a spinal cord injury patient and maybe compare that.
Okay, so so amongst all of our CSF samples, the vast majority are all people living with a spinal cord injury. But one of the things that I did not really discuss is sort of the division and breakdown of those different samples. So we actually do have a sample set, mostly Asia A, Bs, and Cs, but we also do have some Asia E control. So those are non-injured patients. However, a lot of these patients were being seen at the Blessing Spinal Cord Center for the purpose of, I think they were degenerative spinal diseases, so not an acute spinal cord injury. And so we have control CSF in with respect to an acute spinal cord injury. But we always have to be careful when we release these to different researchers to specify that, what might be a control for spinal cord injury may not be a control for something else. And the other thing is that as part of our mission and our vision and our objective, we do try to really reserve these for spinal cord injury research in particular. And the reason for that is just that, especially with those Asia E's, we have so few samples that we want to be able to release those to researchers who are already conducting acute spinal cord injury research, so that we're not wasting those precious controls. And Brian Kwan is interested in trying to collect more Asia E controls in the future. We just don't really have a study proposed yet in order to do that, because as you mentioned, CSF is very, very hard to come by. So.
Thank you for that response. And Caitlin, feel free to follow up with any questions that you may have if anything was unclear. You did mention earlier on in your presentation that you're one of many biobanks in the world. And I was wondering how closely you work with other biobanks in terms of developing SOPs. And I guess in terms of just the number of projects and the number of specimens, this might not be a fair comparison because spinal cord injury isn't as large in terms of prevalence as other conditions. But how does it compare? And so how do you collaborate, and how does your organization compare versus other ones around the world?
Yeah, so like I said, as far as we know, we're the first polyuser spinal cord injury biobank, but there is that international organization called ISBE, so the International Society for Biological Environmental Repositories. And ISBE is sort of the leading network for collaborations in biobanking and also biobanking education. So I served as a committee member for their pediatric special interest group because I had come from a children's hospital biobank. And what ISBE does is, through different conferences, and actually even more usefully is a biobank focused forum that allows biobanks from all levels of staff to be able to ask questions, receive papers and research data and materials, get some firsthand experience. It also allows for job postings and headhunting for different, whether it be specimens or staff. And so what we've done in the last 4 or 5 months on top of our website, which was just opened up in early October, what we've done is we've sort of posted our website on all of these different international biobanking communities, and they have biobank locators that can sort of direct researchers to your biobank and to asking for your samples particularly. And I, having come from Children's Hospital Biobank and having worked there for 5 years, as you mentioned in my bio, Children's Hospital Biobank was the first pediatric institutional biobank in the nation. And what that means is that they're not disease specific, but they're integrated within a hospital. So one of the great things about Children's Hospital Biobank, aside from it being pediatric samples, which is quite rare, is that they collect a wide variety of different diseases and specimen types. So that's where there's a great opportunity to collect normal controls. We used to collect CSF from a lot of leukemia patients, in which they would be collecting CSF to evaluate potential CNS infiltration with blast cells. And if it was negative, well, it was pre-chemo. So essentially that could be used as a healthy control. And so that's a great source for normal CSF if anybody ever needs it, BC Children's Hospital Biobank. And the other thing that was really nice is, even for relatively normal controls, one thing that we sort of established at Children's was to collect discard tonsillar tissue. So a lot of kids get their tonsil tissue removed. Usually that gets tossed. And so we would obtain consent in the pre-op and be able to reserve that, possibly, snap freeze some tissue or extract mononuclear cells from it after, crushing them down and extracting those cells. And so, personally, obviously, I have an investment with with BC Children's that I've had tons of experience there and know exactly what they have available. But aside from that, the International Society for Biological Environmental Repositories is the best option for being able to link and collaborate with other biobanks around the world.
Wow, very interesting. It's like another world that not too many people are aware of. It's fascinating. So are there any last questions from anyone who is here? Okay, I do have one final question for you. Just wondering whether your title as Biobanker, whether that's an official title, or do you have to somehow rise up to a certain ranking or something like that?
Yeah, I mean, I think especially amongst the ISBE and the biobanking community, we all just kind of refer to ourselves as biobankers first. Like I said at the beginning of the presentation, and with that those pillars of biobanks. We have to wear so many hats as a biobanker. And so, first and foremost, I like to consider myself a biobanker before, a manager, before a scientist, whatever you want to call it, just because it's such a unique and niche field that I work in. Yeah.
Excellent. Thank you very much, Adam. That was a very insightful and very interesting talk. Adam's contact is on this slide here, so if you need to contact him with any questions that you may have, feel free to do so. Do you have a few minutes after the talk in case people may have questions that they want to ask after?
Yeah.
Okay, so Adam will be around after the talk as well. Once again, I'd like to thank McLeish Orlando Lawyers again for sponsoring the lunch at Lyndhurst for this month's lecture. Next month, our speaker, we have a special lecture on March 24th at noon that is going to be co-hosted with the Collaborative Program in Neuroscience. And the speaker next month will be Monica Perez from the Shirley Ryan Ability Lab at Northwestern University in the US. This is a co-hosted event, so it's not a sponsored talk. And there will be a link where you do not need to register in advance. So check your emails for advertisement for that talk in the next couple of weeks. Again, I'd like to thank everyone for coming. I'd like to thank Adam for presenting. And just to let you know, the recording for this talk will be available on the KITE YouTube page in the coming weeks. So thank you again, everyone, and I hope you have a good week.